Okay, so we get to talk about chapter 14, which is going to be an introduction to host defenses and innate immunities. You'd think that I'd have this figured out by now, which buttons to pick, but I didn't. All right, so for this one, it's the defense mechanisms with a host. And so the host defenses are going to be innate, natural defenses that are present at birth. They provide nonspecific resistance to the infection. And we also have adaptive immunities. And so these ones we adapt to our surroundings. So they're going to be specific and they're going to be more acquired after birth. So the ones that we're born with are going to be our innate natural ones, where the adaptive ones are ones that we're going to be getting after we're born. So this is table 14.1, and these are general features of the host defenses, and it talks about the first line of defense, which would be your first, second, and third, and whether or not it's innate or acquired. So first and second are innate, you're born with them, and the third line of a defense, you're going to be acquiring it after you're born. And then also the specificity and then examples. So our first line of defense is going to be your physical barriers such as your skin, tears, coughing, sneezing, your pH in different areas of your bodies, as well as genetic barriers such as resistance that are inherent in our genetic makeup. All the way to our second line of defense, which is mostly non-specific. These are phagocytosis, like those Pac-Mans that are going to eat those microbes, right? Inflammation, fever, infection, or interferon. So basically, our body's trying to fight off the bad things that are happening. And then our third line of defense would be things like T lymphocytes, B lymphocytes, and antibodies that we're going to develop as we age and get exposed to different parts of the world. So defense mechanisms of the host are basically to protect the body against pathogens. The immune system relies on a multi-level network of physical barriers, immunologically active cells, and a variety of different chemicals. Again, that first line of defense is going to be any barrier that blocks the invasion at the port of entry, and these, again, are nonspecific. Your second line of defense, like we just talked about, are going to be those protective cells and fluids, including inflammation and phagocytosis, those little Pac-Man guys, and these again are going to be nonspecific. And then our third line of defense, these are the ones that we are acquired with exposure to the foreign substances. These are going to produce protective antibodies and creates memory cells that are specific. So it's kind of interesting because, for instance, I'm growing up here in rural Michigan, right? And so my third line defense would be different than if I grew up in Africa or China or anywhere else, because it's different for our environment. So talking about our physical or anatomical barriers, our first line of defense, like I said, our skin is going to be the biggest one. So your skin and your mucous membranes and the respiratory, urogenital, eyes, and digestive tracts are going to be your first line of defense. This is the outermost layer of skin, and it's composed of epithelial cells, which are compacted, cemented together, and impregnated with keratin. Basically, they're strong. And there are very few pathogens that can penetrate this skin if it's intact. And that's a big caveat, because if you have, for instance, a scrape or a cut, then obviously that first line is kind of like open, right? You also have a flushing effect of sweat glands. We can have blinking and tear production, and also stomach acid that can help to defend these different microbes. And so this is figure 14.1. It's titled defenses. And so these would be some nonspecific defense barriers that help prevent entry of microorganisms into the host tissue. So we also have mucus coats, and these are going to be impeding the attachment and entry of bacteria. So basically, if it gets caught in your snot, it can't get into your respiratory system. And that's what this basically boils down to. Our nasal hairs are going to help to trap those larger particles, okay? And so this is figure 14.2, and this is the ciliary defense of the respiratory tree. And in A, which is on the right, the trachea and bronchi are structured to entrap and propel particles upwards towards the pharynx, where in B on the left, this is a detail of the trachea at 5,000 times magnification, and this features tufts of cilia on the surface of the epithelial cells that sweep away particles and microbes. And so think about this. So I'm a farm girl. We used to have to do hay, right, back in the day. After we did hay, you'd go and you'd blow your nose, right, and it was all gross and cruddy, or you're in the dust, whatever, and it's cruddy. It's because that snot is going to be trying to trap those microbes so that it can't come in and infect your body. We also have non-specific chemical defenses, and these would be like our sebaceous secretions. One really big one is a lysosome, and the lysosome is an enzyme that hydrolyzes the cell wall of bacteria, and this is present in tears. So to lyse means to break apart, okay? And so that makes sense that this would be hydrolyzing. And if you, again, look towards those root words that I've always stressed, hydro is using water, to lyse is to break apart. So a lysosome is going to be in your tears, which have a water content to help break apart any of those bacteria to stop them from getting in and causing an infection. We also have high lactic acid and electrolyte concentration in sweat. So if you guys have ever worked out really hard and you get sweat like on your brow, right, your lips, right, and you taste it, it's kind of salty and bitter. And this is because of that lactic acid, is that bitter taste, and then it's salty because the electrolytes concentration in your sweat. Your skin's pH is also slightly acidic, so that's a natural barrier. You have hydrochloric acid in your stomach, and so our pH of our stomach wall, or stomach in itself, is right around 2, so that's very, very acidic. And this is because of the presence of hydrochloric acid, and that's a very, very strong acid that will help break down things in our stomach. And think about it, we're eating picnic food that might be out and have little bacteria on it, and it'll help to protect you and get those away, okay? You also have digestive juices and bile in the intestines, and semen also contains an antimicrobial chemical. So, and the vagina has an acidic pH, so that helps to keep everything in that homeostatic level so that we hopefully don't get any problems. But when we do, we can fight them off, okay? What about genetic defenses? Well, some hosts are genetically immune to the diseases of other hosts, and some pathogens have great specificity. So these are both playing in our favor. And these genetic differences are going to exist in susceptibility. Okay, so basically we're built for the way we're built so that we can fight off things that we would have to fight off. But like, I don't need to have my cat's immune system because she's going to deal with different microbes, etc. So looking at this and the structure and the function on the organs of defense and immunity, you can see that this study of the body's second line and third line of defense is called immunology. And so this is basically the functions of a healthy functioning immune system. And the first thing we do is survey the body to make sure that everything's intact. If we have a problem, then we're going to recognize that foreign material. And then in the third step, we would actually destruct the entities that are deemed to be foreign. And so you can see here that we survey, we detect, and then we destroy. Okay. With the immune system, this is a large, complex, and diffuse networks of cells and fluids that penetrate into every organ and tissue. There are 4 major subdivisions of the immune systems. The first one is the RES, second one is ECF, third is the bloodstream, and fourth is the lymphatic system. So for the first one, it's reticular endothelial system, and this is also known as your mononuclear phagocyte system, or MPS for short. The second one is extracellular fluid, or ECF, and again, the bloodstream and the lymphatic system. So looking at our immune system definitions, we have white blood cells, and these are called leukocytes. And these have the innate capacity to recognize and differentiate any foreign material. Okay, innate means you're born with it. It just knows what to do. Okay, so it's like danger, Will Robinson, danger, right? There's a foreign body here. We need to figure out what to do with it. We also have non-self, and this is going to be the actual foreign material itself. Then you have self, and this is the normal cells of your body that are supposed to be there. Then you also have pathogen-associated patterns, or PAMPs, and these are molecules that are shared by microorganisms. And then you also have pathogen recognition receptors, or PRRs, and these are receptors on the white blood cells for those pathogen-associated patterns. So body compartments that participate in the immune system are going to be the reticular endothelial system, the extracellular fluid, the bloodstream, and the lymphatic system. So this is figure 14.4, and it's titled Connections Between the Body Compartments. So in A, over on the left, this is the meeting of the major fluid compartments at the microscopic level. The close proximity of these complements at the cellular level allows for the continuous exchange of cells, fluids, and molecules, and that would be the arrows that you can see moving things back and forth right in there. Okay, and then in B over here is a picture or a schematic view of how the main fluid compartments form a continuous cyclic system, or cyclic, making up words again, cyclic system in exchange. So it keeps going in a circle, right? And so the positioning of the compartments favor rapid circulation from one site to another, and it's either through direct or indirect connections. And so the direct is going to be the solid arrows, and the indirect is going to be the dotted arrows. Okay, so again, that's figure 14.4. When looking at the reticular endothelial system, otherwise known as the RES system, this is the network of connective tissue fibers that interconnect other cells, and it meshes with the connective tissue in the surrounding network organs. And so this is inhabited by the phagocytic cells, such as mononuclear phagocyte system, and these are macrophages that are ready to attack and ingest the microbes that pass the first line of defense. And this is figure 14.5 in your book, and it's titled The Reticular Endothelial System Occurs as a Continuous Connective Tissue Framework Throughout the Body. So this system begins at the microscopic level with fibrous support network, which is those reticular fibers enmeshing each cell. The web connects from one cell to another within a tissue or an organ, and that provides a niche for phagocytic white blood cells, and these can then crawl within and between tissues. The degrees of shading in the body are indicative of variations in the phagocytic concentration, and the darker the area means the greater phagocyte ability. The origin, composition, and functions of blood. Well, whole blood is going to consist of plasma and formed elements, or blood cells. The serum is the liquid portion of the blood that forms after a clot has been formed, minus the clotting factors. Where the plasma is 92% water, it has metabolic proteins, globulins, clotting factors, hormones, and other chemical gases to support the normal physiologic functions. And so this is figure 14.6 in your book. It's the macroscopic composition of whole blood. So in A on the left, when the uncoagulated blood sits in a tube, it striatifies, which is just a fancy word for layering, into a clear layer of plasma, a thin layer of off-white material called the Buffy coat. Buffy the Vampire Slayer. Okay, sorry. A Buffy coat, and this contains the white blood cells, and a layer of red blood cells in the bottom. And then in B on the right, you can see the serum is the clear fluid that separates from the clotted blood. The clotting causes the red and white cells to clump at the bottom of the cells or the bottom of the tube in a single mass. So you can see this clotting over here. So when we're looking more specifically at the white or the blood cells, we have hemopoiesis, and this is basically the production of blood cells. You have stem cells, and these are undifferentiated cells, which are precursors of new blood cells. And then you have leukocytes, and these are white blood cells, and you have a couple subsets of those, which would be the granulocytes and the agranulocytes. And the granulocytes are going to be have a lobed nucleus, where the agranulocytes are going to be unlobed with a rounded nucleus. Okay, so looking at this figure, basically this is a version of 14.7 in your book, and so basically this is a simplified diagram of blood cells and platelet development. The details of several intermediate steps have been omitted. The undifferentiated stem cells in the red marrow give rise to several different cell lines that become increasingly specialized until mature cells are released into the circulation. The yellow area, which isn't yellow on yours, but it would basically be like this area. So from the myeloblast and around, this indicates mature leukocytes or white blood cells, and the details of and the photomicrographs of major circulating leukocytes are featured with the descriptions. Okay, so look this up in your book because it talks about the different neutrophils, basophils, and eosinophils, and what they are and the specifications of each. Okay. So granulocytes, neutrophils compose about 55 to 90%. And again, these have a lobed nuclei with lavender granules, and these are phagocytes. So they're going to eat things, right? They're Pac-Mans. We have eosinophils, and these only compose of about 1 to 3%, and these are orange granules and bilobed nuclei. and these are going to destroy eukaryotic pathogens. And then you also have basophils, and this is about 0.5%, and these have constricted nuclei with dark blue granules, and they're going to release more potent chemical mediators. And then of those, we have mast cells, and these are non-motile, meaning they can't move, and these are elements that are bound to our connective tissue. Neutrophils, these are light purple granules in acid-base stain, and this is how they would present underneath the microscope. Again, they have a lobed nucleus. Other names would be SEGs or polymorphonuclear leukocytes, or bands, which are young neutrophils. These are the first to arrive at the infections. They are phagocytic, and 54 to 62% of leukocytes are neutrophils. And they are elevated in bacterial infections. Eosinophils, these have deep red granules in the acid stain. These have bilobed nuclei, and these have moderate allergic reactions, and they defend against parasitic worm infestations. And these make up about 1 to 3% of leukocytes. And these are elevated in parasitic worm infestations and allergic reactions. Basophils have deep blue granules in the basic stain, right? These are going to release histamines, release heparin, and then these compose less than 1% of the leukocyte group. And these are similar to eosinophils in both the size and shape of the nucleus. Then you have granulocytes, and these are lymphocytes, which are about 20 to 35%, and they have a specific immune response. Specifically, and we talk about these a lot, we have B cells and T cells, and the B cells have humoral immunity. And so these activated B cells are going to produce antibodies. So these are pretty important for us. You also have T cells, and these are cell-mediated immunity, and these are activated T cells that are going to modulate the immune functions and kill foreign cells. You also have monocytes and macrophages, and these are about 3 to 7% of the population, and these are the largest of the white blood cells. They have a kidney-shaped nucleus, and again, they're going to be phagocytic. I like how I always put an extra syllable in there. Macrophages, these have a final differentiation of monocytes, and then we have our dendritic cells, and these are going to trap pathogens and participate in immune reactions. Monocytes, these are the largest of all blood cells, okay, and all of these are on my book in page 446. So monocytes have the largest of all the blood cells. They are spherical or round. They have a kidney-shaped or oval or lobed nuclei right there. They leave the bloodstream to become macrophages, and these are 3 to 9% of leukocytes, and they phagocyte or eat up bacteria, dead cells, and other debris. Lymphocytes are slightly larger than our red blood cells, and they have a large spherical nucleus surrounded by a thin rim of cytoplasm. And the T cells and B cells are both important in immunity. B cells can produce antibodies, and these are about 25 to 30% of the leukocytes. So here is a fun summary table. This is table 14.2 in your book of the characteristic of leukocytes. So this is just basically a summary of what we all just talked about, and just put a little star by this because this is going to be a great summary table for you. With erythrocytes and platelet lines, well, erythrocytes develop from bone marrow stem cells. They have a loose nucleus, and they are simple biconcave sacs of hemoglobin. Where platelets, these are formed elements in circulating blood, and they are not whole cells. Okay, so there's this distinct differentiation. Moving on to the lymphatic system, this is figure 14.8. And basically this lymphatic system is going to provide an auxiliary route for the return of extracellular fluid to the circulatory system. It acts as a drain-off system for the inflammatory response, and it renders the surveillance, recognition, and protection against foreign material. So reading the summary here in A, which is the body, okay, you can see A right here. This is a generalized depiction of the lymphatic system, a branching network of specialized vessels that extend to the most regions of the body. And it includes several types of supportive tissues and organs, including the lymph nodes, malt, spleen, galt, thymus gland, tonsils, and bone marrow. where when we look into the expanded view B, then this is going to be the view of the right breast and the auxiliary lymphatic circulation. The right lymphatic duct receives lymph from the right axillary in the head region, and it drains into the regular blood circulation at the right subclavian vein. And on the left side of the body, there is a similar arrangement in which the thoracic lymphatic duct returns the lymph from the remainder of the body at the left subclavian vein. And through these direct connections, all lymph eventually makes its way back into the circulating blood. So what is lymphatic fluid? Well, lymph is a plasma-like liquid that is carried by lymphatic circulation. And it's formed when blood components move out of blood vessels into the extracellular spaces. This is made up of water, dissolved salts, and it's about 2 to 5% protein. And this transports the blood cells, fats, cellular debris, and infectious agents. So this is a close-up of your lymphatic vessels, and this is in figure 14.9. And so basically your lymphatic capillaries permeate all parts of the body except for the central nervous system, bone, placenta, and thymus. The thin walls are easily permeated by the extracellular fluid, which is then moved through the contraction of skeletal muscles. And the function is to return lymph to the circulation, and the flow is one directional. And that means it's going to flow towards the heart, and eventually it's going to return into the blood system. Okay, so again, you can see this is figure 14.10, comparison of the circulation patterns of regulatory circulatory systems and the lymphatic system. The movement of lymphatic flow is one directional, which is the green one, and from the lymphatic capillaries to collecting vessels and the ducts to the large lymphatic trunks to the subclavian veins in the heart. The flow of blood, on the other hand, is cyclic, with blood continuously flowing through the arteries and capillaries and veins to the heart and back around. With this combined system, the lymphatics can collect excess tissues, fluid, and return it to the bloodstream. The two systems can also participate together in the surveillance of the tissues for foreign invaders. Note, the lymphatic flow, which is again green, is shown in only one side to keep the comparison uncluttered. So this is figure 14.9, and this is a picture of circulation in the lymphatic vessels and lymph nodes. And so basically, in A up here, the finest level of lymphatic circulation begins with blind capillaries, which are in the green, and they're gonna pick up foreign material from the surrounding tissues and transport it in the lymph away from the extremities via a system of small transport ducts. And then in B here, the ducts carry the lymph into a circuit of larger ducts that ultimately flow into the clusters of filtering organs in the lymph nodes. Then in C, down here, you can see that this is a section through the lymph node, and it's going to reveal the afferent ducts that are draining lymph into the sinuses that house several types of white blood cells. Here, the foreign material is filtered out and processed by lymphocytes, macrophages, and dendritic cells. And then down here in D and E, the lymph continues to trickle from the lymph nodes via efferent ducts into a system of larger drainage vessels, which ultimately connect with the large veins near the heart. In this way, the cells and products of immunity continually enter into regular circulation. So with this one, this is talking about lymphoid organs and tissues, and this is figure 14.11. So this is classified as primary and secondary, and so basically our primary lymphoid organs are the sites of lymphatic origin and maturation, and these would include your thymus and the bone marrow. Where our secondary lymphoid organs and tissues, these are circulatory-based locations such as the spleen and the lymph nodes, and these collect the cells that are distributed through the body cells and tissues, and this would include your skin and mucous membranes, your salt, galt, and malt. Sounds like I'm ordering a shake to go, right? And so in, again, figure 14.11, this is especially outlining the thymus gland. And so immediately after birth, the thymus is a large organ that nearly fills the region over the midline of the upper thoracic region. In the adult, however, it is proportionately smaller. And this section shows the main anatomical regions of the thymus, where the immature T cells are going to enter through the cortex and migrate into the medulla as they mature. Um, so what specifically are our lymphoid organs? Well, our thymus is one, and has a high rate of growth and activity until puberty, and then it begins to shrink. And this is the site of T cell maturation. You have our lymph nodes, and these are small encapsulated bean-shaped organs that are stationed along the lymphatic channels and large blood vessels of the thoracic and abdominal cavities. Then you have the spleen, and this is structurally similar to the lymph node, and this filters out the circulating blood to remove worn out red blood cells and pathogens. And then you also have miscellaneous lymphoid organs such as Galt and Peyer's patch. So what are the actions of our second line of defense? Well, basically it's recognition, right? Inflammation, phagocytosis, the interferon components, and its complement system. And so specifically talking about our inflammatory response, the classic signs and symptoms are characterized by redness, warmth, swelling, and pain. And so this is figure 14.2 in your book, and it's titled The Response to Injury. And this is the classic series of reactions. And so basically the redness is going to have increased circulation and vasodilation in an injured tissue in response to the chemical mediators. The warmth is the heat that's given off by the increased blood flow. The swelling is going to be increased fluid that's escaping into the tissues as the blood vessels dilate, and this would be like edema, white blood cells, microbes, debris, and fluid collecting to form pus, and therefore preventing the spread of the infection. Then also the pain, you're going to be stimulating the nociceptors or the nerve endings that are sensitive to pain. So looking a little bit more closely at the major events in inflammation, this is figure 14.13, and you can see the red is going to be the rubor or the calor of the inflammation. The yellow is going to be edema or the tumor indolor, and then the blue here is going to be the repaired or damaged tissues. And so basically, if we start up here in A, this is the injury and the immediate reactions, and this is where the blood vessels are going to narrow, causing vasoconstriction. And then blood clots can form, and mast cells can release those chemokines and cytokines. So chemokines and cytokines. Oh my God, I can't talk today. Into the injured area. Okay, so the bacteria can enter into the wound. The mast cells are going to release those chemical mediators, and again, we're causing vasoconstriction. Then we go to B, and these are our vascular reactions. And so this is where the nearby blood vessels are going to dilate, increasing that blood flow, increasing the vascular permeability, and increasing the leakage of fluid forms to form the exudate. And so you have your clot that's going to form, you have your bacteria, you have your neutrophil, then you have your seepage here of the plasma and the migration of white blood cells out of the blood vessels. And again, you now have vasodilation. For C, this is edema and pus formation. So this is the collection of fluid with edema or swelling, and this is when we have the infiltration by neutrophils and the formation of pus. And then down here in D, you have the resolution and scar formation, and this is when we have our macrophages, the lymphocytes, and the fibroblasts that are going to migrate in, initiate the immune response, and repair the injury. It's going to cause a scar, potential scarring, and loss of normal tissue. Okay, so those are going to be your major events in inflammation. The chemical mediators of the inflammatory response. So this is figure 14.14 in your book. And so basically these are the major chemical mediators of the inflammatory response and their effects. And so you have your vasoactive actions. And this is going to cause like your vasodilation, your increased permeability of the capillaries in the small vein, and also vasoconstriction. So it can cause both dilation and constriction. And those chemicals would be like histamine, serotonin, bradykinin, and prostaglandins. You also have the initiating event, and this would be where you have trauma, infection, necrosis, foreign particles, or neoplasm. And these are going to be the ones that are going to initiate that effect. And then you have your chemotactic actions, and these are where cells can migrate to the site of the damage, and those would be like your neutrophils, your platelets, your macrophages, your lymphocytes. You have some mediators with both vasoactive and chemotactic events, and these would be your complement components because they're complementing each other and working together. And some of these would be like cytokines such as interferon and interleukin, and some products of arachidonic acid metabolism, as well as platelet activators. Then you also have substances with chemotactic effects, and these would be like your endotoxins, your platelet activating factors, your leukotrienes, your mast cell chemotactic factors, as well as your bacterial peptides and PAMPs. So leukocytes have some unique characteristics, and one of them would be diapedesis, and this would be the migration of cells out of the blood vessels and into the tissue. And then you also have chemotaxis, and this would be a migration in response to a specific chemical at that site of injury. Okay, so this is figure 14.5. And so this is a cross-section of a venule, which is a small vein, and this depicts the white blood cells squeezing themselves between the spaces in the vessel wall through diapedesis. This process also indicates how the pool of leukocytes adheres to the receptors in the endothelial wall. From the sites, they're poised to migrate out of the vessels and into the tissue space and respond to chemotactic factors that are released as part of the generalized inflammatory process. So what about fever, right? Well, this is initiated by circulating pyrogens, and these reset the hypothalamus to increase body temperature, and this can signal muscles to increase heat production and vasoconstriction. You have exogenous pyrogens, and these are products of infectious agents. And you have endogenous pyrogens, and these are liberated by monocytes, neutrophils, and macrophages during the phagocytosis. And examples of these would be interleukin-1 and tumor necrosis factor, abbreviated TNF. Fevers are good. I mean, they can be a pain in the butt to deal with, but they're actually beneficial, okay? And these basically can inhibit the multiplication of temperature-sensitive microorganisms. It can also impede nutrition of bacteria by reducing the available ions. And it can also increase the metabolism and stimulate the immune reactions and protective physiologic processes. Phagocytosis, well, this generally activates the general activities of phagocytes are to, one, survey the tissue compartments and discover microbes, particulate matter, and dead cells or injured cells, or two, to ingest and eliminate these materials. The third activity that they do is to take those immunologic information from the foreign matter. So say, yo, what's going on? Okay, let's report you to the big guys and get this taken care of. Okay. So phagocytes and phagocytosis. I have always taught these guys as Pac-Mans. Okay. And you guys might be too young to know what Pac-Man is. It was a video game when I was growing up. Okay, so this is figure 14.6. And this would be the developmental stages of monocytes and macrophages. And basically the cells process through the maturational stages in the bone marrow and peripheral blood. And once in the tissues, the macrophages and dendritic cells can remain nomadic, which is relaxed, kind of like in the latent period, right? Or they can take up residence in a specific organ. And so our neutrophils, these are general purpose. They react early to the bacteria and other foreign materials, and also to the damaged tissue. You have eosinophils, and these are attracted to the sites of parasitic infections as well as antigen-antibody reactions. And then you also have macrophages, and these are derived from monocytes, and they scavenge and process foreign substances to prepare them for reactions with the B and T lymphocytes. Okay, so they're very important. So how do we recognize these foreign cells? Okay. Well, this is figure 14.18, and this is phagocyte detection and signaling with toll-like receptors. You have to pay a toll to pass, right? Toll-like receptors, which are abbreviated TLRs, span the membrane of the phagocytes and other cells in the immune system. When a molecule such as PAMP on a particular pathogen is recognized by this receptor, the TLRs merge and bind to the foreign molecule. And this is going to induce the production of chemicals and therefore trigger engulfment. And so protein-like receptors within the cell membranes of macrophages are called toll-like receptors. And these are going to detect foreign molecules and, again, signal the macrophage to produce chemicals to stimulate an immune response. Okay. So what is the mechanism of phagocytic recognition, engulfment, and killing? Okay, so chemotaxis and ingestion, this is when the phagocytes migrate and recognize those PAMPs, and this would be like a phagosome. And then the phagosome lysome is going to be a lysome, which is fused with a phagosome. And this basically is going to cause death of that infection within about 30 minutes. Then you also have destruction and elimination. And this is going to be oxygen-dependent system, and it's called a respiratory burst. And this is where we have the liberation of lactic acid, the lysosome, as well as nitric oxide. So looking at phagocytosis, this is figure 14.17, and this is the sequential events in phagocytosis. So up here in 1, what's going to happen is the phagocyte is attracted to the bacteria. And then in 2, we have a close-up view of the process showing the bacteria adhering or sticking to the phagocyte receptors by their PAMPs. And then 3, that vacuole, which is going to be that little vessel or bubble, is formed around the bacteria during engulfment. And then in 4, the phagosome is going to have digestive vacuole results. And then here in 5, the lysosomes fuse with the phagosomes, forming a phagolysosome. And then down here in 6, the enzymes and toxic oxygen products kill and digest the bacteria. In 7, the undigested particles are released. And then you can see here in the inset, this is a scanning electron micrograph of the neutrophil in yellow, and this is actively engaged in devouring the anthrax bacteria. So he's having a snack, right? He's eating it. He's doing his job. He's doing awesome stuff. What about these interferons? Okay, this is figure 14.9. So small proteins that are produced by certain white blood cells and tissue cells are interferons. And so we have interferon alpha, we have interferon beta, and we have interferon gamma. And so alphas are going to be those lymphocytes and the macrophages, where the beta is going to be the fibroblasts and the epithelial cells, and then the gamma is going to be those T cells. And these are produced in response to viruses, RNA, immune products, and various antigens. And they bind to cell surfaces and induce the expression of antiviral proteins that inhibit the expression of cancer genes. And so this figure again is 14.19, and this is the antiviral activity of the interferon. So when the cell is infected by a virus, its nucleus is triggered to transcribe and translate the interferon gene. Interferon diffuses out of the cell and binds to interferon receptors on nearby uninfected cells, where it produces the production of proteins that eliminate the viral genes and block viral replication. Note that the initial cell is not protected by its own interferon, and that interferon does not prevent recipient cells from being invaded by the virus, but the viruses are inactivated before they can start replicating. Okay, so they're pretty cool. So what is a complement? Well, this is a versatile backup system. So basically, this consists of 26 different blood proteins that work in concert to destroy bacteria and viruses. The complement proteins are activated by cleavage, and this is going to initiate a cascade reaction. And so the pathways are going to be classical, which would be activated by the presence of the antibody bound to the microorganism. You have your lectin pathway, and this is the nonspecific reaction of a host serum protein that binds to mannan. Then you have alternative, and this begins when the complementary proteins bind to the normal cell wall and the surface components of the microorganism. So this next picture is figure 14.20, and this would be an overview of the complement pathways leading to the membrane attack. So in this first part, you have initiation. So all 3 different pathways have the different triggers and starting points, but they all converge at the same place. And that would be here at the C3 convertase. And this enzyme begins the series of reaction, which would then be seen in the B, C, and D. So the next picture in the next slide. Okay. And then these give rise to the membrane attack complex characteristics of the complement machinery. And the final result is the formation of tiny openings in the cell membrane and the destruction of the target pathogen. So again, we have 3 different pathways that are converged here to the C3 convertase, and this converts the C3 molecule to an activator of the cascade, which is C3b. So cascade amplification is happening here, and basically this is the C5 factors acted on by C3b, and this converts it to C5b. And then C5b becomes bound to the membrane and serves as a starting material for the chain of events that are going to be shown in the next picture. And so C3A, it's not showing it, but this guy, C3A and C3-5, have additional inflammatory functions. Both molecules stimulate mast cell degranulation, and they enhance the chemotaxis, or the chemical moving, of white blood cells, and they act as inflammatory mediators also. All right, step 3 is the polymerization, where C5b is a reactive site for the final assembly of an attack complex. In the series C6, C7, C8, they're all going to aggregate with C5b and become integrated into the membrane. They form a substrate upon which the final component C9, okay, which is this blue guy here, can bind. Up to 15 of these C9 units ring the central core of the final membrane attack complex, or abbreviated MAC, okay? Membrane attack complex, MAC. And then then 4, we have the membrane attack, and so this is the insertion of those MAC the insertion of MACs, and this is going to produce hundreds of tiny holes in the cell membrane, and this can cause cell lysis and death of eukaryotic cells and many gram-negative bacteria. All right, so this is the final picture in this series of slides. And so this is figure 14.21, and this is the flow chart summarizing the major components of the host defenses. So defenses are classified into one of 2 general categories. 1 is innate or nonspecific, 2 is acquired or specific, and these can be further subdivided into the first, second, and third lines of defense, each being characterized by different levels and types of protection. The third line of defense is the most varied, and it's responsible for specific immunity. And this is covered in greater detail in chapter 15. All right, thanks, guys.